Okay, so today's session is for your laboratory session, and we have done three experiment in online mode. And before start moving to the next experiment, so have you finalized your decision for coming or not coming, or still it's
So some of us are coming, some of us are not coming.
okay? So those who are coming, so for them next week we are going to have a physical practical. So whatever you have, whatever I have shown you, or I have described you that one is the extraction of DNA. So you are going to extract your DNA from cauliflower, and after that you are going to quantitate that also. So that that quantitation you are going to use your spectrophotometer. So these both the experiment you are going to do physically while you when you are going to join your lab. And the third one is that I describe your construction of vector. So construction of vector is a practical that you can do simply on online. So there is you need just a laptop and some online tools that you need. So what are like restriction site finders that you all have. I think you all know from your previous semester. So that just for that you don't need any physical type of thing. Just I can give you some assignment, so you can do that one. Okay, so now we are moving to our fourth experiment, and before moving to the fourth experiment, I will just visit your lab records, what you have submitted, and that three things that you have to submit. One is your uh, isolation of DNA that you all have done. So I have returned you also. And the second is was a quantification of DNA. So out of 23, 20 people have done something. So I'm checking that and I'm returning that to you. And so Adish, so here Ishita is late this time again. So she's 2 day late. So she will mark 0. And IT sport won't need another
So
Nandini
actually, this
this time
time
also.
I was unable to. No, so actually, last time I have submitted on time, but this time there was some issue with my MS Teams. I
See,
was
it was
unable to
7 days. So it's I think I think 2 days
So
I
yes, it's for 2 days. Yes, sir. So one day it was not showing at my end. The next day when I operated on another device, I borrowed laptop from my cousin, and in that it was operating. That's why I'm late. Sorry, that's my fault.
these are
Sorry, sir.
just excuses. So
No,
last time I have given everyone marks for those who were late, but now no excuse. So Ishita is late, and second is Nandini again. This time also Nandini is late. So Nandini will mark 0 this time. And third one is Vivek Gupta. So Vivek Gupta didn't upload it yet, so he will also be marked 0 this time. Okay, so these are the students who didn't did, and I'm returning you with your marks.
Excuse me, sir.
Yes.
Actually, Vivek is facing continuous network issue, as well as he's been in and out with hospital and family issues. That's why he's facing some issues, and he's just texted me right now. So I'm just trying to inform you.
Okay, for today we may think about that, but for the assignment part, uh, no excuse. And one more is the Rupal Joshi. Rupal is also didn't submit it, so Rupal is also marked 0. So just a moment. Okay. Okay, students, I have just checked your assignment box. You got marks. And third assignment timing is tomorrow, 12 o'clock. Okay, so I hope you all have written the writing part, and only the things that remained is the uploading. Okay. Okay, so now we are moving to our today's one, and today we are going to talk about our fourth experiment. That's in, I think all of it is visible on all of your screen. And this experiment is going to talk about isolation of your plasmid DNA from bacterial cell. As in our previous class, we were talking about vector, and vector. We mentioned that this is a plasmid is a vector, and we can use this plasmid as a vector. And now question is, we want this vector alone. And as in our first experiment, when we were talking about the isolation of DNA, when you isolate your DNA means you take out, when you lyse your cell, everything is going to come out. Means your genomic DNA and also the means your chromosome. When you say the genome means both plasmid and your chromosomal DNA. So here the more precise distinction between these two type of DNA is one the chromosomal DNA you can say, and second is the plasmid DNA or extra chromosomal DNA, that is also plasmid DNA. Okay, so as I mentioned that both are the DNA, so chemically, physically, or their by their property there is no difference. Both are going to have deoxyribose nucleotides, so nothing, and both are going to be double stranded. And in case of bacteria, both your chromosomal DNA and your plasmid DNA both are circular also, so they are going to be supercoiled also. So everything, every protein is going to be same for that. So no big change is going to be there. Okay, so now simply I want to ask you have how we can get just plasmid DNA. Just give some idea. Let's say by this way we can this property is different. In this way, the plasmid DNA is different from the chromosomal DNA. So this difference between the property we can utilize for separating them. So anyone can give any idea, any suggestion? Any difference between them? So you might be all like I think you are in the final year of your course, so definitely you all are aware about what are the difference between your plasmid DNA and your chromosomal DNA. No response? A, a deep silence?
Sir, so the difference between plasmid DNA and chromosomal DNA is that plasmid DNA can replicate on its own, and chromosomal DNA uses, certain mechanisms and enzymes.
For the replication wise, both are replicating in similar way. Same way, not at similar, in same way. Whatever machinery you are referring for the chromosomal DNA, same required for the plasmid DNA also. Both are replicating their own, requiring all the enzymes that you, all the polymerases or the helicases, ATPase, whatever enzymes that you need for the replication of your chromosomal DNA, all the machinery that you required for the plasmid DNA also. So, but for the sake of replication, there is no difference.
Sir, chromosomal DNA replicate with genome, whether the plasmid DNA replicate independent of the genome.
Yeah, that is, but how this replication property will help us in separating these two DNA? Other than this,
Sir, plasmid is double stranded, whereas DNA, chromosomal DNA can be single or double.
Demetrius, please give me example of any bacteria which have a single stranded DNA. It will be a novel discovery if you mention a name of bacteria which have single stranded DNA. Yes, Demetrius.
No idea, sir.
Then you mentioned that chromosomal
I was
DNA
talking
can be
about.
single stranded also.
I wasn't talking from a bacterial point of view,
Then
like
whose point of
normal
view you are talking?
chromosomal DNA, normal.
Whose chromosomal DNA is single stranded? Name an organism whose chromosomal DNA is single stranded.
So I'm not sure.
Oh, you are giving the example you are citing, so you must be sure. That's why I'm saying your name must be nominated for Nobel Prize.
Thank you,
That
sir.
discovered an organism which with the single stranded DNA. Except viruses, except viruses, no organism have single stranded DNA. Okay, so DNA is always double stranded. And okay, so this is one thing. So any other differences? Any other differences?
They are extra chromosomal. They are not required by the cell to survive. They just provide extra specific functions to the cell.
Yeah, that's fine. But is this property is going to help us in means a cell which is not having a plasmid DNA? Then we can't isolate a plasmid from that cell.
Sir, no idea.
Means when we are going to isolate a plasmid, that means that cell must have a plasmid. Then only we are going to isolate. So now we are not talking about cell are having it or not having it. Here the simple question is between plasmid and chromosomal DNA of a bacteria. Means you have to consider that cell is going to have plasmid. We are referring a cell which is having plasmid and also having a chromosomal DNA. So we are talking about that cell. So how these two things are there that cell is having, how these are going to be differing from each other. Okay, so I'm giving you a 5 minute break. So please think, give a deep thought on the differences. And after 5 minutes, we'll join. I will come back to you and we'll continue our discussion. Okay, students. Now welcome back. So now, yes, Yuvraj.
Sir, I had submitted my assignment on time, but still I can see the marks is 0/2.
Just a minute. प्रतिमा. Now you can check it again.
Now it's fine.
It's fine.
Yeah.
Okay, so now you have to just not to focus on the marks. You have to focus on also on the question. So answer what I'm asking. Yes, Yuvraj. Difference between the chromosomal DNA and the plasmid DNA of a bacteria. Yes, Yuvraj. After getting marks, you also become silent. I gave you people 5 minutes. You can Google in this much time. And after Google, you can check the differences. What are the difference between the chromosomal DNA and the plasmid DNA? Even simple this thing you people can't do. Yes, Shakti.
So can we separate them on the basis of their size? Like plasmid DNA are smaller than chromosomal DNA.
Yes, you are well said. One is the size. So this is the main difference between the plasmid DNA and the chromosomal DNA. Chromosomal DNAs are very large in size, wherever plasmid DNA is comparatively very small in size. That's the one difference. So on the basis of size, we can do that one. And one more property we can utilize in the separation of your DNA, plasmid DNA. That is the in our yesterday in our previous class, I think on Friday we were talking about the property of your plasmid DNA. When we were talking about just a moment. When we were talking about the topoisomerase, and what is that property? When we were talking about the topoisomerases. When you're talking about the topoisomerases, we talk about as a property of your circular DNA. Yuvraj, turn on your mic first, and if you don't want to speak, keep your mic off. Yuvraj. Yes. So, what are the property that we talk about the topoisomerase? In our previous class, we talked about the topoisomerases, and at that time, the action of topoisomerase, we were talking about some property. And this is the property in which your topoisomerases are involved. And some of you also asked why, yes. Yes, Yukta, you want to say something, Yukta?
Yes, sir. Yes, sir. You explained that with the help of topoisomerases, we can change the topology of the DNA. Means they're responsible for in winding and unwinding of the DNA.
Okay,
We
what is
can
that?
change.
What is that topology? What we
Sir,
call that?
we doesn't require an extra enzymes. Like topoisomerase can also cut as well as ligase the enzyme, ligase the vector.
I'm not asking the property of topoisomerase. I'm asking the property of DNA.
Property of DNA.
Yes, Yukta, you were talking about the topology. So, what we call to that topology? Yukta?
Um, Yes, sir. I was saying it affects the winding and unwinding, like changing the direction. We talked about.
To that what we call, your mic is off, Ekta. That winding and winding that winding and unwinding is simply your double helical structure you're talking about. But what is that property when we are referring?
Um,
Some negative and the positive you are calling at a time.
so, yes, sir. Negative and positive supercoiling, I think.
Yes, that is the right word,
Yes.
supercoiling. So these are the two property on the that basis we are able to separate our plasmid DNA with your chromosomal DNA. First property is the size, and the second property is the supercoiling. So what this how this supercoiling is going to help you. So supercoiling means they are in a like this is one strand and two strands, and when they're supercoiled, they are entangled in this way. Means you can say simply these two rings they are crossing each other. So it's your DNA is in this way. So in single stranded, it is going to be like this. But when it is a circular one, this is single stranded, this is again single stranded. But when they form double stranded, not just they are coming very close to each other in this way, they are coming in this way. Okay, they are entangled because of this supercoiling when they were in the double stranded form, and these strands they shift over each other. That as we were talking about the topoisomerase, that when it is going below or it is coming above. So there's that second property that called the supercoiling. So this supercoiling make your two DNA strands stay together even when they are single stranded. Okay, wherever the supercoiling is not there, in the single stranded form, these two strands they are going to be away from each other or at a distant location. Okay, so what is going to happen? What it is going to make? What a difference it is going to make? So difference it is going to make that when you want to make it again double stranded. Now it is single stranded. If they are very close to each other, if you make them double stranded, they will become quickly, very fast. But when these strands are away from each other and now you want to make them double stranded, they are going to take a bit time. So they will take time when to come close together and form a double stranded one. So this is the thing that I am going to refer you. This is the one. These are some properties that we required to separate your chromosomal DNA from your plasmid. Okay, so how we are going to utilize it? So we are going to utilize it the method that we call the alkaline lysis method. What we are going to do in that one? In that one, simple thing we are going to utilize term alkaline is there. So what do you mean by the alkaline? What do you mean by the alkaline?
When I use both these sequences and involves, it is aligning them.
What do you mean by the alkaline? So alkaline is simple, I think, from the uh, eighth class. I think you're talking about the alkaline things. Alkali,
Having pH more than 7.
it's having pH more than 7. It's basic pH simply.
Basic.
And this basicity is because of what? This basic pH is because of what? Can you give any example of any base? Yes, NaOH. NaOH. So in NaOH, this basic nature is because of sodium ion or hydroxide ion.
Hydroxide ion.
Hydroxide ion, OH. Okay. So why this your double stranded DNA is double stranded? Why your two strands they are held together? Yuvraj, speak near to the mic. Your voice is very faint to me.
I'm audible, sir.
Yes, now you are better.
So, sir, is it like that the two strands are attached because of the bonding between hydrogen bonding between them?
Yes, you are right. Hydrogen bonding between them. So now next question is, what is hydrogen bond?
Sir, the single hydrogen molecule.
Single hydrogen molecule. Means between two double-stranded DNA, hydrogen gas is there. You want to say this? Yes, Yuvraj?
So it's basically a bond between a proton and an electronegative atom.
Proton and electronegative atom,
Yeah,
HCl. HCl. Hydrogen is linked to the chloride. So this one is the hydrogen bond. Yes, Demetrius.
so that's the covalent bond.
And you said that the electronegative element and a proton is a hydrogen bond. These things all you have studied in your chemistry, I think. Dr. Jasleen have taught you very nicely. She is a very good teacher. You are insulting her, not telling about saying hydrogen bond. And you are also insulting not just Dr. Jasleen, you are also insulting your other teachers also. I think definitely they also talk about this hydrogen bond. You are also insulting your school teachers also. Your school teacher who taught you in the 11th class, this hydrogen bond is there in 11th class, I think in the chapter number 5, bonding. You're insulting them also. You're also insulting your under 11th, 10th or 9th teachers also. So how many teachers you're insulting?
But I guess like you're confused because I said an like an attraction, electrostatic attraction. Because HCl में the electrons are shared right between. That's the HCl is an example of covalent bond because the electron is shared between them.
So give me the
Whereas
example of a hydrogen bond.
what's the
Give me the example.
of hydrogen so in H2O water.
So H2OH. So oxygen and hydrogen bond. This is a hydrogen bond.
So there is and
Hmm?
intramolecular hydrogen bonding,
Intramolecular.
and there is
So in water, the intramolecular means within the H and O is the intramolecular.
so I guess you're not getting my point. There is when the electrons are shared, then it's covalent bond. But when there is an electrostatic attraction between them from one hydrogen and another molecule, then it's
So, how this
hydrogen bonding.
electrostatic attraction is going to result?
So I just said cause of the electronegative atom.
Okay. So the chief is now saying bit better way. Okay. So this hydrogen bond is yes,
So
Marisuni.
if I
Yes, Marisuni.
Sir, so I think what he was trying to say is that it's the interaction between a hydrogen atom and any atom which is more electronegative than hydrogen. So any atom which has a higher affinity for electrons. So for example, let's take in the case of DNA itself, the hydrogen bonds are formed between the bases between in one purine and one pyrimidine, right? So that interaction is also between hydrogen and either oxygen or nitrogen, since both atoms have a very high affinity for electrons. So it's not sharing, it's just an interaction between the two.
Okay. So you also said something, but not also I think able to describe it completely. So simply just school definition I'm telling you. So school definition is that when hydrogen is covalently bonded. So now this bond is covalent. Okay. When the hydrogen is covalently bonded with highly electronegative element. Okay. So what I'm saying, hydrogen is covalently bonded with highly electronegative elements. Now what are these electronegative elements? Your chlorine, oxygen and nitrogen. When hydrogen is bonded covalently, again I am repeating this term covalently. When this hydrogen is bonded with this covalently, this highly electronegative element, this results in development of partial negative charge on electronegative element and partially positive charge on the proton. Okay, so this gives a development of a polarity over this molecule. So now the hydrogen is partially positively charged and nitrogen is partially negative charged. So now this partially positive charge can attract another partially negative charge nitrogen, chlorine and oxygen, or vice versa. So this electrostatic force of attraction between these two means your hydrogen, and condition is hydrogen should be not just should, hydrogen must be bonded with highly electronegative elements. So these are the three one, means FON, chlorine, oxygen and nitrogen. Then this hydrogen is going to have a partially positive charge. This partial positive charge can attract a partial negative charge over another partially positive proton. Simple as the Demetrius were talking about the water. Yes, in case of water there is a hydrogen bond, but this bond is intermolecular. So here the term is not intramolecular. So term must be intermolecular between two molecules. So now the hydrogen is bonded with oxygen, that is an electronegative one. This is going to result in development of partial positive charge over the hydrogen. Now this partial positive charge hydrogen can attract a partial negative oxygen of a second water molecule. So this electrostatic force of attraction between your partially positive one water molecule and partially negative another water molecule is called hydrogen bond. So condition is there, hydrogen bond, that electronegative must be, hydrogen must be bonded with the electronegative element. So your nitrogenous bases, they are having NH and OH or O. Okay, so this NH and O, so H is going to be partially negative because it is bonded with the N, and O is going to be, or N is going to be partially negative because they are electronegative. Okay, so because of that, these two can attract such type of bonds that you are calling the hydrogen bond. So now as I want to introduce you this OH, the alkyne one. So this alkyne one OH is also going to contribute, also going to interfere with this hydrogen bond because it is going to contribute to your O, that is going to be negatively one, and H, that may be going to be a positive one. Okay, so this OH hydroxide ion, they are going to interfere with these hydrogen bonds that are present between two nitrogenous bases, between two DNA strands. When they interfere with this hydrogen bond, what they are going to do? They are going to break the hydrogen bond. When they break the hydrogen bond, your DNA, which was earlier double-stranded, now your DNA is going to become single-stranded. Okay, so this is the purpose of your alkaline, the term that I mentioned in your method, the alkaline lysis method that we are going to use. We are going to use some alkali. What this alkali is going to do? This alkali is going to interfere with your hydrogen bond and will make your double-stranded DNA to the single-stranded. Okay, so now again, as we are talking about the differences between your plasmid DNA and the chromosomal DNA, both are DNA, so both are going to have a hydrogen bond, so both are going to become single-stranded. Okay, so in the alkaline condition, when your DNA is there, your plasmid DNA and your chromosomal DNA, both are going to become single-stranded. So now nothing is going to change whether they are going to be single-stranded form or the double-stranded form. Okay, so what we want to do, we want to separate them. So now the next step that is going to help us in separating them, that is, again we will make them double-stranded. So how to make them double-stranded? So now how to make them double-stranded again? So as I mentioned, introduction of the alkali, make them a single-stranded. So simple thing, you will say that, sir, remove the alkali. If you remove the alkali, it will become again the double stranded. So how to remove the alkali? How we can remove the alkali? How we can remove the alkali? Yes, Maniswini? Yukta?
So we can neutralize it by addition of acid.
Yes, well said. We can neutralize by addition of acid. Okay, so this step is called neutralization. So first we make the alkaline condition that make the single strand, double stranded and single stranded. Now you are adding the alkali. Sorry, you are adding the acid. So when you add the acid, your single stranded again become the double strand. Okay, so now what game is there in between these two? So now the game is there that your plasmid DNA, very small, being supercoiled, it will form double stranded bond. Now DNA, which was very big in size, very large in size, when you make neutralize it, it will also start forming that double stranded. But being it's larger in size, and also one more thing is there in your chromosome. When you define your chromosome, you mentioned that chromosome is nucleoproteo complex. Means protein is also there in your chromosomes. So now these proteins and single-stranded DNA, they will entangle with each other. Okay, so like now instead of forming a double stranded, it is going to entangle with your protein. Okay, but that's going to lead the precipitation of your DNA and protein. Wherever in case of your plasmid being smaller in size, it will become double stranded, and this double stranded form going to remain soluble. Where the chromosomal DNA being larger in size, being entangled with the protein, got precipitate out. So here not just you are mentioning that just this entanglement is going to precipitate. So main thing is that this protein when you neutralize, neutralization also make your protein to precipitate. Okay, so with this protein precipitate, large DNA form a complex and it also precipitate down. Okay, while plasmid DNA being smaller in size and also being supercoiled, they are very near to each other. They will form double stranded one and a soluble one. So they are not going to precipitate out. Okay, so these two things, one thing that the alkaline is going to make your double stranded single stranded one, then neutralization is facilitating making single stranded to again double stranded. But during this alkaline and neutralization, your chromosomal DNA and protein will precipitate out wherever your plasmid DNA again form double stranded one and remain soluble. So this is the two things, two property. One is the size, plasmid being smaller size and chromosome being larger size. So chromosome will precipitate, plasmid won't precipitate. Plasmid being smaller in size and being supercoiled immediately form a double stranded and will remain insoluble wherever your single stranded one DNA with the pure proteins they won't form the double stranded one and it won't remain in the solution and it is going to precipitate out. So this is the principle of your plasmid DNA extraction by simple that method that we call the alkaline lysis method. So the principle where you can see, and under the alkaline condition, your double stranded DNA forms a single stranded, and when you utilize it, your single stranded DNA, plasmid DNA being smaller size, it forms the double stranded one and remain soluble. Wherever DNA being larger in size, it forms complex with the protein and precipitates out. Okay, so this is a simple way you can say this is a simple principle of your plasmid DNA extraction. Okay, so what method we are going to follow when we are saying we are going to isolate your DNA, some solution are going to be there, some compounds are going to be there. So then some cells definitely need which are going to have a plasmid. So all these things we will discuss in our next class. And now I'm resting this session. And please turn on your camera to record your attendance when I call your name. So just turn on your camera And in next session, we'll start first with the discussion and followed by the description, detailed description of the protocol and the requirement, what we need and how we are going to proceed. During that, I will also show you some video that will tell you how you are going to proceed while you are going to do experiment practically. Okay, so I'm calling your name for attendance, so please turn on your camera while I call you. It is Adish. Adish.
Present, sir.
Atoiba. Atoiba.
Present, sir.
Atoiba, why you was absent in your previous 2-3 classes? Atoiba, why you was absent?
Hello?
Yes, Atoiba, why you was absent in your previous classes?
Sir, there was network issue, that's why I can't log in.
Okay, so next is Ayushi. Ayushi done. Demetrius.
Present, sir. Present, sir.
Devanshi. Devanshi. Ishita.
Present, sir.
Janvi.
Present, sir.
Kshitij. Manaswini.
Present, sir.
Namrita.
Present, sir.
Nandini.
Present, sir.
Nehal.
Present, sir.
Rupal. Rupal.
Present, sir.
Sahiba.
Present, sir.
Sanjana.
Present, sir.
Shakti.
Present, sir.
Shivani.
Present, sir.
Tanvi.
Present, sir.
Tridev. Tridev. Vivek. Yukta.
Present, sir.
Yuvraj.
Present,
So
sir.
all are present today. So today is great. So today all are present. So still we have 5 more minutes. So any question before ending this session? If anyone of you would like to ask, please let me know. Yes, Vivek, I noticed you are present. Any questions? We have still 5 minutes to end the session. कोई सवाल नहीं? So no questions. Keep asking questions, because if you keep asking, then when I am going to ask, then you can answer it. Because if you don't ask, then doubts will remain with you. Okay, and then you will give you a wrong explanation of the things.
So do this and
Okay. Yes, Yuvraj, you want to ask anything?
I'm listening. Yes, sir.
Okay. So stay safe, stay happy. We'll join back after 5 minutes.
before we start the next one,